Good morning, good afternoon, good evening. Welcome everyone to today's Global IMMUNOTALK. Global IMMUNOTALKs are an initiative created by Dr. Elina Zuniga and Carla Rotlin in 2020 to spread immunology and inspire the new generation of immunologists all around the world in a friendly and egalitarian manner. I am Gabriel Rabinovich and have the honor and privilege of presenting our today's speaker, Dr. Michael Lenardo. Mike received his BA from the Johns Hopkins University in 1977 and an MD from Washington University in 1981. He then undertook clinical and research training at the University of Iowa and was a postdoctoral fellow at the Whitehead Institute at MIT under the supervision of David Baltimore and Philip Sharp. He became second-chief in National Institute of Allergy and Immunological Diseases at the NIH in 1989, investigating T lymphocyte regulation, HIV-1, and genetic immune diseases, and was named NIH Distinguished Investigator in 2019. He has published more than 300 articles and has filed several medical patents. Mike is a pioneer in the field of immunoregulation and cell death. He discovered the propacidal mechanism of immune regulation and defined several genetic immunological diseases. He led special efforts to have 2 drugs approved by the FDA in 2023. His honors and awards include his recognition as a member of the National Academy of Sciences, National Academy of Medicine, and the American Academy of Arts and Sciences. He was also awarded with the American Association of Immunologists Tayman Award and the NIH Director Award, among many others. On a personal note, I would like to say that I was almost going to be a postdoc in Mike's lab in 1999 with a Pew Fellowship. Unfortunately, due to family reasons, I could not leave Argentina then. So I always wish to have the opportunity to express my admiration for Mike and thank him for his unique generosity. Thank you so much, Mike. Mike will talk today about the impact of genomics on human immunological diseases. But before you start your talk, Mike, one of the traditions of Global IMMUNOTALKs is to ask special questions to inspire the young generation of scientists. So the question today for you is, could you share with us one of the most impactful decisions in your scientific career?
Yes. When I was looking for a postdoc at the end of my training in Iowa, I had several offers for different laboratories. And for a variety of reasons, that mainly revolved around science, I chose to go to David Baltimore's lab. And that was a very, very important decision. And I think illustrates something that's important for young people, and that is to go to a lab where the lab leader is a person who really knows how to think about science and think about important experiments. Because what that will do is change the way you think, as it did for me, about how to think about picking important scientific questions and pursuing them successfully. So I think, I guess it boils down to mentorship, choosing the best mentor you can avail yourself of, and really working hard when you're under them to get all of the learning and knowledge that you can from that experience.
Okay, thank you very much, Mike, for these very inspirational words. And now I would like to invite you to share the screen and to deliver your lecture.
Great. Thank you, Gabriel. I really appreciate your very generous introduction. And let me see if I can put this in presentation mode. Okay, and this is a little bit ahead of where it's supposed to be. Okay, so as Gabriel said, I'm from the National Institutes of Health. This shows a picture of our hospital. My lab is in the hospital complex. And over the years, although I was mainly trained as a basic scientist, I began to look at different types of clinical diseases, mainly genetic diseases of the immune system. And that's what I will talk to you about today. So to do that, what's shown on the left side of this slide is what I call the virtuous circle of discovery. And that shows that you start with a clinical workup, work through modern genomic technologies, and then go back to basic biology, essentially biochemistry and molecular biology, to understand what the gene that you're studying that's mutant in the patient that you're studying, what the impact is. And then you go back into the clinic for genetic counseling and hopefully, if you're lucky, a new therapy. And one thing I want to emphasize, which is shown on the right side of this slide, is that the work that we have done has been collaborative around the world. We have 2,600 referrals over the last 18 years from all over the United States, Europe, even some from Asia. And these have been really exciting collaborations. The 4 centers shown here are all in the country of Turkey. And these have been our most enthusiastic participants. Our team has gone over to visit these centers. We've worked very closely with them and co-published with them some of the exciting diseases that we've studied. So global interaction, global collaboration is really central to what we've done in our program here at NIH. So I'm going to go through 2 diseases and try and illustrate how studying the genes will illuminate pathways that are absolutely crucial for the immune system to work properly, and also lead us to concepts for treatment of these diseases. So in this case, it was a case of patients that came in with immunodeficiency, infections, and this was due to the loss of lymphocytes during their lifespan, usually within their childhood. It was also associated with liver disease. I won't say a lot about that in this particular presentation, but it accompanies the other problems. And they also have signs of autoimmunity, anemia, and splenomegaly. And these pictures here just show, and let me turn my cursor here into a laser pointer, shows the very large splenomegaly. This is the lower rib margin here, and you can see it extends much greatly below that, almost into the pelvis. And this shows the evidence of severe bronchiectasis and due to infections. There's actually a huge infection right there. And this also shows some of the liver disease that's in these families. And so in these 9 families, we found that there were loss of function biallelic deleterious mutations in a protein called Gmap5. And this protein encodes a small GTPase, which is this domain here in the protein, but it also has a hydrophobic segment that can be involved in anchoring it to membranes. And just a spoiler alert for what I'll talk about at the very end, the membrane that it chooses to go to mostly is the outer membrane of the lysosome. So you can see these families in many cases are consanguineous, as shown by the double line between the 2 parents. And you can see it's also not a very benign disease, because many of the children that have been in our study have actually passed away, and it has been due to this disease. So the consequence of the mutations is that they lose the Gmap5 protein. Gmap5 is actually part of a family of genes that are all highly related and linked in the genome, but the disease is specific for Gmap5 and does not affect any of the other genes. When you look at these genes, you can see they all, everyone in the family has the same basic structure with a GTPase domain at the N-terminus, and then either coiled or hydrophobic segments on the C-terminus. Gmap8 is exceptional in that it has 3 kind of triplication of these regions. And these are unique among GTPases in that they don't work by gaps and GAFs, which control GTPases like RAS, but rather they are regulated by homo and heterodimerization. Now, these are conserved in evolution. They seem to be the result of a duplication and divergence event, and they end up in different parts of the cell. So other Gmaps go to other places, and they apparently have other functions. Gmap5 in particular is very highly expressed in T cells and endothelial cells. This seems to relate to the phenotype of the patients that we have found, in that it affects immune function and also the liver through endothelial cells in the liver. And I won't, as I said, I won't go into that. Now, it turns out Gmap5 is actually a pretty famous gene even before we started working on it. in that it is the gene that is knocked out, you might say, by a homozygous frameshift mutation in the very famous biobreeding or BB rats, which are a strain of rat that was picked up in research decades ago that develops type 1 diabetes, but can also develop other autoimmune diseases depending on the genetic background of the rat. And then later it was picked up in an ENU screen in mice done in Bruce Beutler's lab, in which the mutation caused T and B cell lymphopenia. It turns out Gmap5 in mice is also in B cells, as well as autoimmune disease and liver disease. So looking very much like the patients. And they worked on it and actually named the mouse Sphinx, which is interesting because they couldn't really figure out what it did, what the gene did biochemically, but it also turns out to be a very relevant moniker if you remember the riddle of the Sphinx, which has to do with aging, human beings aging. And you'll see why I mention that as I get further into the talk. So now a variety of groups have worked on this, and there were a number of different theories out there as to what it did, but the underlying function was clearly not, there was not a consensus on what it did. So we started to look at it, and this is a very busy slide, but I'll walk you through. It's actually quite simple, but it shows a couple of very important features of the lymphocytes that are left in the animal. As I said, most of the lymphocytes, most of the T cells go away. So first of all, what you can see is in the upper quadrant of cells stained with CCR7 and CD45RA, the cells in the right upper quadrant that would normally be considered naive are really depleted quite severely in the patients in both CD4 cells and CD8 cells compared to the controls. And all these graphs are just these done for every single patient that we studied in our different families. And this graph at the end, which I'm pointing at with my cursor here, is just a summary of that. Now, we, because it looked like there was activation in more effector memory cells, we wondered, since the cells didn't seem to be very functional in other data I'm not showing you, we wondered if the cells had been overactivated and then gone into senescence. And so a nice marker for that is CD57. You can see the three patients here in the colored trace all have high levels of CD57. Again, this graph just shows each individual patient that we studied, and here's a summary of those. And it turns out that, and this is true again for CD4 and CD8 cells, but that was not the sole marker. Once we found that we wanted to look at, we actually looked at a number of other markers of senescent cells, KLRG1, the senescence-associated beta-galactosidase, and then two markers that are decreased in senescent cells, CD27 and CD28. And then also, senescent cells are known to hypersecrete certain panel of cytokines, including IL-6 and IL-8. And you can see those are both increased. So when you look at all the data together, it looks like these cells have had some sort of an abnormal hyperstimulation and gone into a senescent state. Now, the cells that we got in the patient, those results were all on patient cells. We now wanted to say, well, what if we go back to the knockout mouse, or the Sphinx mouse, which was actually an ENU mutation that led to loss of function, and see whether other characteristics of senescence are there. And some of the ones we were particularly interested in looking at were markers of cell cycle control, because that could account for the loss of cells over time. And so if you look at these three graphs, P21, P53, and P16, all of them are elevated in the Sphinx mice compared to the controls. And interestingly, if you look at mice that are 16 to 19 months old, that we would consider aged mice, you can see that they're also elevated there. And again, here's what I showed you for the humans, the senescence-associated beta-galactosidase, and that's increased in the Sphinx mice as well as in the old mice. Now, one of the problems that we thought about was the fact that because these cells are so unusual, when we compare them to a normal mouse, it's sort of like comparing apples and oranges. These cells are clearly, they don't look like naive cells. So we wanted to know, is this truly due to a direct effect of Gmap loss, or is this just something about cells sitting around in an abnormal mouse that makes them look like this? And so in order to do that, we activated cells from a normal mouse and then did a CRISPR-Cas9 knockout of Gmap5, and then looked a few days after that at what the behavior was of those cells. And what is very strikingly clear, and I don't show you all the biochemical markers that I showed you just now, but rather look directly at cell viable cell number. As the knockout it progresses, you can see it drops precipitously in the knockout mouse, and it's not a form of apoptosis, but rather it's the breakdown of the membrane in a form of necrosis. So this says that just the removal of the Gmap5 gene in otherwise normal cells, you get a phenotype that mimics what happens in vivo and is likely due to senescence leading to some sort of death event. And the death event is unusual in that it's not a program of cell death. So this then led to a whole variety of experiments, and it wasn't until we turned to Erica Pierce to help us do metabolomics and lipidomics that we actually began to understand what is going on. And what she found was a striking increase in ceramides, which are normally kept at very low levels and tightly regulated. And I'll tell you more about ceramides in just a second. And there were very specific kinds, usually long-chain ceramides and hydroxyceramides, but not dihydroceramides. And so this was very characteristic of the patients. It was not found in other unaffected family members or parents or controls that were from the same location, the same city. And so this really, to us, was a big clue. And this just shows on the right-hand side the kind of schematic of the ceramide metabolism pathway that obviously is closely related to sphingolipids and sphingomyelin. So ceramide, as you can see, is just sphingosine linked to a fatty acid residue, and this residue can be of variable length and have variable numbers of single and double bonds. And the role of ceramides in cells are sort of twofold. On the left-hand side, you can see they actually are signaling roles, and not surprisingly, they can cause senescence and cell death. But they're also involved in very low quantities during T cell activation, in proliferation, and also in some cell types in differentiation. And they can also, on the right-hand side of the slide, you can see they play a structural role in the cell, but they're usually present in very, very controlled amounts. And when we looked through the literature, we found that dysregulation of ceramide levels has been implicated in all sorts of aging and age-related diseases, including inflammation, neurodegenerative disorders, type 2 diabetes, and cardiovascular disease. So that goes back then to the riddle of the Sphinx, that if you remember the different types of walking on 2 legs, 3 legs, and 1 leg, 4 legs, 2 legs, and 3 legs was related to the age of the person at different stages. And so we thought it was very interesting that we found a gene, a pathway that was involved in control of senescence of cells and related to changes that happen in older mice. So if you look, again, this is now an acute knockout of Gmap5 in otherwise normal cells, and you can see just knocking out that gene alone is sufficient to cause the accumulation of long-chain ceramides, and that's shown here on a heat map. And so, so we really felt that we had found the true regulatory role of Gmap5. So this is an experiment that Casper Hobie, the postdoc in Bruce Beutner's lab, who identified the Sphinx mice, did that I thought was very revealing for how the production of ceramides is controlled. And what he did was he crossed the Sphinx mouse to an OT2 mouse, and he showed that the mice and the T cells are normal as the mouse grows older until he puts ovalbumin into the drinking water. And at that point, Hi, Gabriel, are you still there?
Yes, I am here.
Okay, so that was the woman from San Diego.
Okay, no, it's perfect because I told her that it was shut off, so she's calling you, but now you can continue.
Okay, so you may have to edit out the part.
Yes, we will cut.
Okay, thank you. So let me start again here. I apologize for the equipment problem. So this shows an experiment that I thought was very revealing for how ceramides are controlled in T cells. And this was done by Caspar Hobie, a fellow with Bruce Beutler, who identified the Sphinx mouse. And what he did was he cleverly crossed the Sphinx mouse to an OT2 mouse and showed that the T cells in the mouse were fine until the mice were fed ovalbumin. And at that point, then all the characteristics of the Sphinx mouse emerged with the loss of T cells and the abnormal phenotype of T cells. And so this implied to us that what was driving the production of ceramides, the excessive ceramides, when they aren't unregulated by the loss of Gmap5, that this was due to T cell receptor stimulation. And what you can see in the graphs in the middle and on the right, the heat map on the right, that there's a very large accumulation of ceramides when you stimulate a Sphinx mouse cells with anti-CD3 and anti-CD28. And so this really suggested that the abnormal survival and viability and phenotype of the cells was due to the overexpression of ceramides. But to test that directly, we needed to block the production of ceramides and then see whether or not that would correct the viability defect. And it turns out there is a ceramide synthase inhibitor named fumonisin B1. And when you use that, you can see, if you compare here the black open bar, which was fumonisin itself, which is a little bit toxic to T cells, and compare that now to Gmap5 knockout cells, you can see there's a drop in viability, and that drop is reversed when you use fumonisin. And also, if you look for the production of ceramides, again shown by this heat map, the production is reduced. So I won't have time to go into all of the background biochemistry that we did to further elucidate the pathway. But in essence, what we found is that the T cell receptor stimulation drives a complex that actually includes Gmap5, LCK, and another kinase called CK2. And when that complex normally exists, then it will lead to CK2 combining with ceramide synthase and producing small amounts of ceramide that are probably involved in T cell receptor signaling. But if Gmap5 is left out of the first complex, we now find that there's a strong constitutive association of CK2 and ceramide synthesis with a huge increase in ceramide. And it turns out if you use a blocker of CK2 called CX4945, you can actually prevent the accumulation of ceramides, showing that this is critical to the overproduction of ceramides under these circumstances. Now, it turns out that along the way, and I mentioned this at the beginning of the talk, we recognized that Gmap5 and probably this complex is being assembled on the surface of the lysosome. And we also found that it can associate with the so-called regulator complex that regulates mTOR. And what you can see is if you do immunoprecipitations of LAMPTORs, which are part of that complex, LAMPTOR1 or LAMPTOR4, you can pull down Gmap5. And so this suggested that rapamycin, which blocks mTOR, might have some role in regulating this complex. And indeed, if you look at the addition of rapamycin, you can see that various senescence parameters are arrested by rapamycin. And we have also tried this in patients. So this is one patient that was given a rapamycin. You can see the size of the spleen was reduced, the lymph nodes were reduced, and the phenotype in the blood seemed to reverse to some extent. But it wasn't a complete reversal of disease. So we're still looking for other interventions in this pathway to help the children that have this disease because it is so severe. So we call this disease Gmap deficiency with infections, splenomegaly, erythrocyte autoantibodies, lymphopenia and liver disease, or Giselle disease, and it's due to recessive loss of function Gmap alleles causing loss of protein and a loss of normal regulator function on the lysosomes. You can diagnose it by reduced Gmap staining or, of course, a genetic investigation. And preliminary data shows response to sirolimus, but we're looking with ongoing studies to other small molecule inhibitors. So now I want to go to the second disease that I wanted to share with you. And this is a disease that we studied for a number of years, in which mainly children referred to us from one of the Turkish centers directed by Ahmet Ozan. And they came into the immunology clinic because they had recurrent infections. And one of the things immunologists do, clinical immunologists, they look at the level of antibody in the blood. And what you can see here in red, the level of IgG was dramatically reduced from the normal level, which is shown in the dotted lines. And in addition, they were surprised to find that all the other proteins in the blood are also reduced, including shown here in black, albumin. So this is a condition called hypoproteinemia. And we also heard from the mothers of these children that they had essentially chronic diarrhea from birth, and they also had chronic abdominal pain, upset, nausea, vomiting, were very reluctant to eat food on some occasions. And so that resulted in us carrying out a gut biopsy of the bowel. And what you can see here are these large stained structures in the gut wall. And these are just very exaggerated, distended lymphatics. And they can be stained by markers that stain lymphatics in the gut. So this condition is called lymphangiectasia, and it's due to destruction and abnormalities in normal structure of the lymphatics. Now, as a consequence of these problems, the children were effectively starving. And you can see these are the normal height and weight charts that you would see when you take a child to a pediatrician's office, and you can see they're way below the lowest decile. And furthermore, the loss of an anticoagulant protein, protein S in the blood, led to further complications, including large clots forming. This is the right atrium of the heart, which can throw off emboli, lethal emboli into the lungs. And this is a massive clot in the lower leg vein, which also can lead to cutting off circulation. So they had all sorts of complications. And this disease was so severe that normally children don't live past the second decade with this condition. So when we carried out genomics on a number of patients with this disease, again, we found a biallelic loss of function mutations now in a gene called CD55. And this is a cell surface protein that has been known for a long time. It used to be called decay accelerating factor. It's a cell surface protein that blocks the complement pathway, which I'll tell you about in just a second. And if you look carefully at these pedigrees, again, you can see a number of the families, in fact, all the families here, the two parents are related, which is shown by the double line. They're usually cousins, and all the children have homozygous configuration by inheriting the same allele, but from each parent. Since this is a cell surface protein, you can stain cells and look for it in the blood, and you can see that these patients have very severe loss of this protein, and this is shown also on a Western blot. So complement originally was described as a series of proteases that are triggered by antibody-coated pathogens, usually bacteria, which are detected by the first complement component, C1q, and they lead to the production of a multi-subunit complex called MAC, or membrane attack complex. And it was also has been known for a long time that some of the split products of these proteolytic reactions, such as C3b, C3a, and C5a, can interact with specific receptors on immune cells, T cells, macrophages, and others, which are called anaphylotoxin receptors, and trigger all sorts of changes in the behavior of those cells and the secretion of cytokines and other immunological properties. And furthermore, we now know that other things that we've heard a lot about in the last couple of decades, damage-associated molecular patterns, apoptotic cell-associated molecular patterns, and pathogen-associated molecular patterns, which are picked up by other C1q-like molecules, can also feed into this pathway. So this is really a part of innate immunity. And the way that CD55 works is it simply binds the C3 convertase and stops the pathway above C5. Now, what does this have to do with the severe gut disease that I described to you? Well, if you look in the guts of children with this disease, you now can stain for very stable split products of complements, such as C3c, and you find it on the surface of the lymphatics. And even the membrane attack complex you can see on the lymphatics. And the consequence of that, the production of anaphylotoxins, there's massive infiltration of inflammatory cells, which are all this purple nuclei here. And this below on the lower right here just shows a massively distended lymphatic, which breaks down and releases that protein-rich lymph fluid into the gut lumen rather than returning it back to the circulation. So that's what's leading to the hypoproteinemia. Now, of course, because of the severity of this disease, we really wanted to do something about this. And so it turned out at the time we did this, there was one drug called eculizumab that was available on the market. It was used for another complement disease called paroxysmal nocturnal hemoglobinuria. And there were also some test compounds like compstatin that had not been used clinically, but were available. And so when you use those, either compstatin or eculizumab, you can see they are very, very potent at flatlining complement activity. And so we had hopes that these could work very well in the clinic. So this is a test that we did in Istanbul with a number of patients there. This was carried out by our collaborator Ahmet Ozan, in which we could now look at total protein, albumin, IgG, and vitamin B12, other macronutrients, before and after the administration of eculizumab, which occurred at time 0. And you can see the axis here is in years. So these patients have had this condition essentially their whole life, and it doesn't get better on its own. And so when you now give them eculizumab, what you see is just a spectacular recovery of all these parameters into the normal zone. And you can see now the axis we're talking about here is literally in weeks. So within a couple of weeks, even less than a couple of weeks, these parameters will go into the normal range. And also, it's very gratifying when you give these drugs to these children, they also immediately lose all of the terrible symptoms that I talked to you about, which include things like abdominal pain, which can be very severe, even requiring narcotics, the nausea, vomiting, and diarrhea. All that vanishes as soon as the complement activation has been arrested. So I just want to show you what this actually looks like in a real patient. This is patient number 5. And you can see here he is very, very sickly. If he's a 15-year-old boy and weighs only 27 pounds. So if I pulled back that blanket, which we didn't do for this photograph, you would see essentially just a raw rib cage with no fat or muscle whatsoever. And you can see, though, in his face, in his head, that his hair is very sparse, the skin is very mottled and not very good. That's a sign of starvation, as well as his dentition is very bad. So we estimate at the time this picture was taken, which was back in 2017, he was literally probably weeks away from dying. And so he received treatment, and after a couple of years of injections of eculizumab, this is what he looks like. So you can see it really, really has a dramatic effect on the physiology of these children. And they go back to living normal lives where they're eating normally, going back to school, playing with other kids, and growing into adulthood. So one of the problems with that drug is it was one of the most expensive drugs on the market and wasn't widely available around the world. So that led us to try to bring another drug onto the market. And luckily, we found that Regeneron, a drug company up in Tarrytown, New York, had a human C5 inhibitory antibody. Also, it was a new drug, had not yet been tested in a clinical trial. And it had the advantages that it could be given by subcutaneous administration, did not require an infusion in a hospital. So it could be given in the home by a parent or actually by the patient themselves. So we set up an open label single arm trial a couple of years ago. We actually brought this boy from rural Bolivia, where he was found to have the disease, to the NIH Clinical Center during the pandemic. And he lived actually on the NIH campus in a facility called the Children's Inn, which is a beautiful home that they have for children that come to NIH for clinical therapy. And you can see here he is with his sister, who came along and spent the 2 years of the trial with him on the NIH campus as his legal guardian. And so this trial was completed last year in May. 1 year ago, the data was locked. And we were and so this shows some of the data we got. And you can see it was very, very spectacular. It had all the same clinical effects. This just shows serum albumin of the 10 patients that were in the trial here. And you can see this is in hospitalizations. You can see they effectively all stopped. This one that happened in patient 1001 was actually unrelated to the disease due to CD55 deficiency. So we were very glad when in August of last year, actually we received word that the FDA approved this drug as the first FDA-approved treatment for children and adults with what we call now CHAPLE disease. And because this occurs in 10 countries, at least 10 countries around the world, Regeneron's working very hard with an international pharmacy to make this drug available to kids around the world. So it's been really exciting to see going from clinical disease all the way through to a new medication. So we call this a disease CD55 deficiency with hyperactivation of complement, angiopathic thrombosis, and protein-losing neuropathy, or CHAPLE disease. It's caused by abnormal complement activation, as I showed you, due to autosomal recessive loss of function mutations in the CD55 gene. And you diagnose this by reduced CD55. You can also use other measurements, but it's very easy to diagnose. And in vitro and clinical studies show that C5 inhibitory antibodies are highly effective treatments, and the clinical trial has been successful. So I think the thing I want to leave you with is that gene identification and scientific investigation, basic fundamental molecular, cellular, immunological investigation, can lead to now new etiologically driven precision medical interventions. And this is not the only example of this from our clinical genomics program here, and I'm sure it's true for other genomics programs around the world. We've had 2 new FDA approved drugs, the one I told you about, and then another called Joenja, which was for another disease that we worked on called APDS, or Pasley disease. But we also found that more conventional, already available drugs such as sirolimus and abatacept have worked for other genetic diseases. So I think this is a very fruitful way to learn more about how the human immune system is regulated, but also actually help patients that have these inborn errors of the immune system. So of course, you can't do this without a large number of very, very talented co-investigators from the NIH team. Alex Lenny Green and Ann Park worked on defining the Giselle disease with Ping Zhang and Li Jinzheng as support. Helen Matthews was our nurse, Morgan Similek is our genetics counselor, Yu Zheng is our bioinformatics, and Helen Su is my collaborator, co-PI here at NIH. She's a board-certified pediatric allergy immunologist and runs her own very successful program of genetic diseases. And Gugu Uzel has been very helpful in the setting up of all the Turkish collaborations that I described to you. And CHAPLE disease is a multi-center collaboration also, and Drew Comrie and Ahmet Ozen worked on this disease at the NIH, and Heather and Ivan also, sorry about that. led to carried out the clinical trial. And so I want to thank you for your attention. I apologize for the technological mishap in the middle of the talk, but hopefully you'll find this talk useful. So thank you very much.
Okay, thank you very much. Thank you very much, Mike, for this fascinating and inspiring work from the clinics to the lab and back again from the lab to the clinics. It's a really inspiring, and this is a wonderful talk as well. So thanks so much. I am sure there are full of virtual applauses for you. And so thanks so much.
Thank you, Gabriel, for the invitation, and thank you for hosting this. I really appreciate it.
So as usual, this talk will be followed by questions and answer on Twitter, X, and I will now share a slide with instructions.
Okay. Okay, I guess I'll log off now.
Okay, so first search for account Global IMMUNOTALKS, then find tweet that says ask questions for Dr. Michael Lenardo here, and then reply to that tweet with your question and mention hashtag Global Immuno. So I would like to finally say good night here in Argentina. And
good night here too.
Probably also in the US.
Yes.
At least in the East Coast. And we look forward to meet you next week with a new Global IMMUNOTALK speaker, Dr. Karina Bortolucci. So thanks so much, and thanks for joining us.
Bye-bye.